Hello everyone, and welcome to the Thermo Fisher Scientific webinar series on Sanger Sequencing and Fragment Analysis Made Easy, corresponding with the launch of Applied Biosystems' new Seq Studio Genetic Analyzer. Today we present the fifth topic in the series, a complete workflow for human cell line authentication, presented by Dr. Steve Jackson, a product applications specialist for Thermo Fisher Scientific. I'm Susie Valdez, and I'll be your moderator for today's educational webinar presented by LabRoots and sponsored by Thermo Fisher Scientific. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on that green Q&A button located at the lower left of your presentation window, type your questions into the box that appear on the screen, and we'll answer as many questions as we have time for at the end of the presentation. Also, notice you are viewing this presentation in a slide window. To enlarge that window, just click on the screen icon located in the lower right. If you have any trouble hearing or seeing this presentation, click on the support button at the top right of the presentation window, or use that Q&A button and let us know you're having a problem. I'd now like to introduce today's speaker, Dr. Steve Jackson. Dr. Jackson has over 10 years of experience in genetic analysis technologies. As a senior member of the genetic analysis R&D team at Thermo Fisher Scientific, he helps develop solutions for the research and healthcare markets, leveraging varied technologies such as capillary electrophoresis, digital PCR, next-generation sequencing, and microarray analysis. Please join me in welcoming Dr. Jackson. I will now turn the presentation over to him. Welcome, sir.
Thank you, Susie, and thank you everybody for joining us today. I'm going to talk about cell line authentication and human sample authentication. I'm specifically going to talk about the solutions that Thermo Fisher has available for making sure that the cell lines and your human samples are exactly what you think they are. So, next slide, please. So, cell line authentication is basically confirming that the cells that you have growing in your culture dish are what you think that they are. This is usually done by comparing to databases such as ATCC, the DSMZ in Europe, or the RECON databases in Japan, and making sure that the tests that you do for the cells that you have growing in your culture are either matching with what you have previously established, or they match up with what these databases say. It's important for when you establish the provenance of new cell lines. For example, when you make a new tumor cell line in your sample dish, you passage it for a while. You want to make sure that at the end of that passaging time that what you have is the same as what you started off with. So establishing the provenance of new cell lines is very important. Authentication is also important when you want to track human tumor xenografts in mouse models. And this is making sure that the tumor that you have is derived from the human cell and it's not from a mouse. So cell line authentication, or the solutions I'm going to present to you, are important for tracking the xenografts in mice. And finally, for some of the ex vivo research applications that are coming up, induced pluripotent stem cells and CAR T research, you want to make sure that the differentiated or the manipulated cells that you have will match up with your donor before they are reintroduced into the donor when doing research for these ex vivo cell therapies. Again, doing cell line authentication, human sample authentication, is extremely important for making sure what you have in your dish or in your test tube is what you think that it is. Why is this so important? Well, it turns out that there are over 500 cell lines that have been misidentified, and over 700 publications have been made with this one cell line, this MDA MD435 cell line. It was originally described as a breast cancer cell line, but upon further examination, it was later determined to be a melanoma cell. This was done by expression analysis, and they found that it wasn't expressing breast epithelial markers, it was expressing melanoma markers. So there are over 700 publications that are made with this cell line that have been misidentified. People have been using this as a model for breast cancer for a long time, and it just is not correct. So that's just one example out of many that are currently being dealt with the research community right now. It's a very big problem, and it's getting to be so big that NIH and several publications are requiring a cell line validation approach when submitting grants or submitting publications. A growing list of publications are shown here. These journals are requiring that they have provided some kind of authenticity test for the cell lines they have, and preventing you from publishing results that are resulting from cell lines that have been misidentified. Again, the list is growing, and it is going to be a large requirement in the future as the problem is becoming more recognized. So the way that the cell lines are most commonly authenticated, or human samples are authenticated, is using a DNA profile. This is derived from forensic methods that are used to differentiate different individuals based on their genotypes at highly variable loci. Illustrated here are some of the loci on the different chromosomes. Either the allele name, like TPOX, FGA, CSFPO, or the more arcane designation of D2S, 1338, etc. These are highly variable loci that can be used to make a molecular fingerprint for individuals and for individual samples as well. These loci are based on short tandem repeats, or STRs. These are microsatellite regions of about 2 to 5 nucleotides that are repeated several times in tandem. They're highly polymorphic. They don't really change the gene expression function, but they do basically provide a way to tag what is present at a particular locus. The allele length is determined by the number of repeat units. What is shown here is one allele has 7 repeats, and the other allele has 8 repeats. These can be analyzed by using labeled oligonucleotides that flank the locus, and provide, after amplification, different sized fragments that can be used to see what is present in the sample. Thermo Fisher Scientific has developed a couple of different methods for doing this with highly multiplexed PCR, looking at between 16 and 24 different loci in a single time, and this provides a highly specific fingerprint for individuals' samples. It's also standardized methods that's useful in forensic analyses and is part of human identification. Whoops. So the way that it works is that you start off with an allelic ladder. I apologize for this. This is a screenshot from one of our experiments where each one of the different grayed out bars shows a different fragment length. And in this allelic ladder, what we have are different peaks that correspond to different fragment lengths. For example, in this first locus here, we have a variety of different peaks that range from small to larger sizes. The peak size then is corresponding to different number of repeats in that STR locus. We have a separate locus, which is a little bit larger fragments, and again, we can then match up the number of peaks, the size of the peaks that are present in this sample with what is known from the STR profile. We also have 3 other different dyes, which would provide a different family of peaks for a total of 16 different loci with a large number of different peaks for each locus in this particular kit. To identify the cell lines or authenticate the cell lines, it's a fairly simple task to basically run the same multiplexed PCR and compare the peaks that you get in the PCR with what you got in the allelic ladder. Again, here we're showing this D4S, I think, whatever the designation is here. There's a single peak at this one particular size, indicating this for this individual at this locus, it's homozygous for the STR that has a allele length of say 13. In this case here, at this particular locus, this individual is heterozygous because they have 2 different sized peaks, indicating that there's 2 different sized STRs at that particular locus. So by going through and using the different loci, then you can generate a cell line profile that is unique for that particular cell line. So Thermo Fisher Scientific has 2 different types of kits available for doing this. We have the Identifiler series and Global Filer. The Identifiler series consists of Identifiler Plus and Identifiler Direct. These are 5 different dyes that query 16 different loci. These cover all of the loci that are used at ATCC, RECON, and DSMZ for checking the authenticity of the cells that you're querying. We also have a kit called Global Filer. This is based off of 6 different dyes, having 24 different loci that provides an extra level of discrimination. Now, these extra loci that are present are outside of what ATCC provides, but the kit provides extra sex determination loci, and it provides a higher degree of specification since there are more loci that are being queried, and is therefore excellent for building up your own database of new and novel cell lines. So the Identifiler Plus and Global Filer kits both can be used for cell line and human sample authentication. For the Identifiler Plus kit, it's based on Purified DNA. The workflow is shown here at the top of the screen here. You start off by isolating genomic DNA using your favorite kit. Of course, Thermo Fisher Scientific has options for isolating purified genomic DNA. You then amplify using an Identifiler Plus kit, run the samples by fragment analysis on Seq Studio or one of our other capillary electrophoresis instruments, and analyze using Gene Mapper. Now, to show what how this works, I basically purified genomic DNA from 5 different cell lines and analyzed them on Seq Studio on our 3500 instrument and 3130 instruments. I used varying amounts of DNA, ranging from 3 nanograms down to 0.1 nanograms. And what I want to point out from this is that using purified DNA, if you use too much of it, you can wind up getting reduced efficiency because of there being excess peaks and spurious peaks that are present. The Identifiler Plus kit is meant to use 1 nanogram or less, and here we see that for 1 nanogram on 3130 instruments, we get a perfect calling frequency, and down at 0.3 nanograms, 300 picograms or less, we get perfect calls. On Seq Studio, we wind up getting out of range errors and spurious peaks because of the extra sensitivity. And using the Seq Studio instruments, using less than 1 nanogram can certainly be tolerated and provide accurate results here. So 1 nanogram is recommended. We also recommend doing a PCR cycle number test to make sure that you are within the range and not producing out-of-range errors by using between 27 and 29 cycles of PCR. The Identifiler Direct kits make use of these nucleic cards. These are cards that have a DNA stabilizing matrix built into them. So all you have to do is basically spot the cells onto these cards, as shown here. Let the cells dry on the card there, and take a small punch from where the cells have been spotted on the card, and analyze the punch directly without having to purify genomic DNA. These were made to be used for blood samples, but they work extremely well using the cell line growth mediates. So in this workflow, what you do is you take your cells, you spot them on the nucleic cards, amplify the spot directly using Identifiler Direct, analyze by fragment analysis, and Gene Mapper software. So to show how these were done, I harvested cells in PBS. I spotted 100 microliters of the PBS solution onto these nucleic cards. I took 1.2 millimeter punches of different dilutions and analyzed them using the Identifiler Direct kit. And what you can see is that by using about 5 times 10 to the 5th cells, undiluted cells, we get a perfect calling of the alleles in these different cell types. By using a 10-fold dilution, we're still able to call a vast majority of the cells on Seq Studio and 3500. But we start to get some dropouts in the 3130, and when you use 100-fold dilutions, we're getting a small number of alleles that are detected. So we recommend that you spot at least 5 times 10 to the 5th cells onto these CoPan nucleic cards for using Identifiler Direct applications. So one of the things that the kits can allow you to do is detect whether or not you have contaminated cells here. So what I'm showing in this case here is a HeLa cell line that was mixed with this M4A4 GFP cell. if you know what the profile is for, say, for example, these M4A4 GFP cells, you can recognize which alleles are coming from the HeLa cells and are not part of what the M4A4 GFP profile is. This allows you to say that if you're seeing 3 peaks, 4 peaks in multiple loci, it's possible that the cell lines that you are using have become contaminated with the second cell line. So to show how this works then, what I did was I took different amounts of HeLa cells, mixed them with the M4A4 GFP cells in PBS, spotted them onto the Copan cards, dried them, and then analyzed punches after drying overnight by PCR. And so you can see here that if on a 50% mixture, a 1-to-1 mixture of HeLa cells and M4A4 GFP cells, you're getting a perfect detection of the HeLa cells, all the HeLa cells alleles that are present in that mixed population. Even at 20% HeLa cells, a 1-to-5 mixture of HeLa cells, you're able to detect the vast majority of the HeLa cell specific alleles that are present. But even down at 10% and 1 to 10 dilution of the HeLa cells into the M4A4 GFP cells, you can still see that over half of the allele specific for HeLa cells can be detected on the 3500 and Seq Studio instruments. So these unique alleles can be detected in around 10 to 15% of a population, but with highest confidence you can tell which cells are contaminating if they're present around 20% of the population in your dish. Now, using purified genomic DNA, you can go down even lower. In this case, I spiked in HeLa purified DNA into M4A4 GFP DNA down to 1%. And even at 1%, I was able to detect about 1/5 of the HeLa cell-specific loci that I expect here on Seq Studio. So what this is saying is that over half the of HeLa cell-specific loci can be detected at 4% of the contaminating DNA. Even down to 1% of the DNA, you can still see HeLa-specific loci, and point to the fact that your cells might be contaminated with a different cell type than you expect. So one of the characteristics about cells grown in culture for a while is that they are genetically unstable, and you can wind up having peak sizes that are different sizes. This is a little bit different than in forensic analyses, because in forensic analyses, if a sample is homozygous or heterozygous, you expect there to be peak sizes of equal representation. In cell lines, though, because you can have this genomic instability, you can see that you have peak heights that are different representations, indicate there's different abundances of cells that have different STR profiles present. So as long as you see this from the outset, you can identify that the cells that have different peak heights that might have arisen from genomic instability. So to show that this is the case here and that the kit is working effectively, I took 1 nanogram of genomic DNA from HapMap samples and from cell lines, analyzed them using Identifier Direct Kit on 3500, and I calculated the ratio of the peak heights that are present at each particular locus. What you'd expect is that if a sample is perfectly heterozygous, that you'd get a peak height of 1, or if the sample is homozygous, I just defined that as being 1. So what I'm showing here is that we have 16 different loci. In this case, I'm looking at amelogenin, and here you can see that in the samples that are HapMap on this side of the green line, we're getting peak ratios that are exactly 1.0. But in the cell lines that are on the right of the green line, you can see that there are ratios that are quite a bit different from 1.0. And this is true for the different loci that are present here. For example, here, in this case here, we again, the HapMap samples on the left of the green line are showing about 1, but the cell lines are showing ratios in this case of about 2, indicating that there's a duplication in HeLa cells at this particular locus. So the different cell lines, there's another example here where this particular locus is quite variable in the cell lines, but again, pretty uniform in the HapMap samples obtained from individual. So allelic imbalance is detectable using these kits. It's likely the result of passaging the cells from many lines in vitro, and is more evident in cell lines than in samples derived from humans that have not been passaged in culture. Okay, so in addition to being able to analyze cell lines and authenticating cell lines, these kits can be used for authenticating human samples and other types of experimentation. So in this case here, we just want to verify that a sample derived from an individual is indeed matches to the donor. In this case here, what we've done is taken buccal swabs buccal swabs from an individual, and T cells from an individual, putting them onto the nucleic cards and getting a profile of the alleles present in these two different samples. And you can see we're just showing one of the channels here from the global filer kit here, and you can see that in the blue channel that we get a perfect match of the T cells to what we got from the original buccal cells, giving us confidence then that the T cells actually came from the individual in question. Another way to look at this is by making sure that induced pluripotent stem cells match up with the donors. In this case, we collected blood samples from donors. Stem cells were collected from the blood samples. They were cultured and differentiated. And then we collected DNA from the original blood sample collection and from the induced pluripotent stem cells. We then randomized, blinded the stem cells, and our collaborator sent them to me for analysis. I took 1 nanogram of the DNA and analyzed them using Identifiler Plus, and calculated an allelic score, which is just on the most general case. An allelic score was simply the sum as all allelic numbers that are present in the sample. And then in this small set, I was able to match allelic scores from the induced cells with the original blood samples here. So, for example, here in this individual, or in this case here, we had an allelic score of 415.2. Again, that's just the sum of the allelic numbers for each one of the different loci present here. And I was able to match the blood to the iPSCs for that individual based on the similarity of the scores here. Here's another example here, another collection from a different individual here, giving a different allelic score here. And again, I was quickly able to match up the iPSCs with the blood from that individual. In this case here, we had 24 different donor collections, different blood and iPSCs derived from those individuals, and they matched up perfectly. So one thing I want to say from this is that the allelic score is a good measure of matching up individuals on a small sample set. But of course, when you look at larger sample sets, you can get allelic scores that are the same, but they're derived from different allelic numbers at different loci. So in larger data sets, you might want to look at a different way of analyzing the allelic score other than just a gross sum. So we're often asked, when is the best time to authenticate? So we recommend authenticating when you're establishing the provenance of a new cell line. When you're just starting off an experiment at a very low passage number, for example, you want to take an L card of those cells, generate a profile of those cells before you do anything further. And this is especially useful when you want to compare profiles of a normal to non-cancerous reference. From the get-go, from the start, you will be able to have profiles. You can quickly see whether or not the cells are what you think that they are. It's also important to authenticate when you get an existing cell line from either a collaborator or from one of the cell line repositories that are available. We know that some of the cell lines that are present in these repositories have been misidentified. We want to make sure that a cell line you get from an individual or from a collection is authentic, and so you'd want to make a profile as soon as you get those cells in, again at a low passage number to make sure that they are correct from the start. We would also want to generate profiles over the course of your workflow to make sure that the cells are remaining what you think that they are over the course of your work. it'd be a good idea to, after each passage number, to take an aliquot of these and again validate that they are genuine and they match up with what the initial profile was. And again, when you get a cell line, you might want to get a baseline profile of your cells before continuing or before pursuing other experiments, so that in the end you can test the cells in the very end and and make sure they match with what you generated at the very beginning of your workflows. So to conclude here, what I've shown you is that short tandem repeat sequences can be useful markers for authenticating human cell lines. I've alluded to the fact that the Seq Studio is provides greater sensitivity relative to 3500 and 3130 platforms, and that simply means that you can use less DNA or fewer PCR cycles relative to the other platforms on a Seq Studio instrument. The Identifier Plus kit can be used with as little as 300 picograms of genomic DNA to authenticate cell lines, and as little as 5 times 10 to the 5th cells per mil of the suspension can be used with the Copan nucleic cards and Identifier Direct kit without having to purify genomic DNA from your cells. And finally, I've shown you that contaminating cells can be identified if they make up at least 20% of the cell culture population. But unique alleles can be identified from contaminating cells in as little as 1 to 5% if you're just looking at single unique alleles in that mixed population. You might not generate a complete profile, but you can detect contaminating alleles in some cases as little as 1% of the population. So finally, I'd like to thank Dr. David Yoder for his help in generating the cell line workflows and the data and the need for authenticating cell lines. I'd like to thank Kamini Varma and her team for helping out with the work described in this presentation. And we also want to emphasize that the products are not sold for cell line authentication in China, Hong Kong, or Japan. They're only used for forensics and prepared use in those countries. So thank you very much for your time, and we'd be happy to answer questions now. Okay, so let me go to the questions that we have received. So one of the questions that we have gotten was whether or not the methods described here will work for frozen cells and for FFPE tissues, and the answer is yes. Fortunately, the identifier kits and the global filer kits were designed to work with forensic samples, which are often highly degraded DNA, and so the amplicons and the loci that have been chosen are specifically used for looking at degraded DNA in forensic samples, and therefore quite applicable to FFPE samples and other examples where the DNA from your cell lines have been degraded or are less than perfect quality. So yes, the global filer identifier kits and the workflows presented here will work with FFPE samples. Another question that we have received is that the Seq Studio that we've talked about in this presentation, what are the benefits of the Seq Studio? And there's people that want to know a little bit more about what it does. So this instrument is a new personal capillary electrophoresis instrument, if you will. It is designed to be extremely easy to use. We have completely redesigned the user interface so that you can set up runs, access data, and analyze the runs quickly and easily. We've designed it to be extremely easy to use in that there's, instead of having different arrays and polymers and buffers that are needed to be added to the instrument, everything is contained within a single cartridge. The cartridge is a simply plug and play, and so you can put the cartridge in the machine and immediately start using. This provides advantages because the cartridge itself is stable on the instrument and at 4 degrees for 4 months. So you can use the same cartridge over and over and over again and save it for quite a while. It also allows users to swap out cartridges so that different labs don't have to cannibalize each other's resources, if you will. The other thing is that we have made the Seq Studio to be network friendly from the get-go, so that you can access the Thermo Fisher cloud and on remote devices, control the instrument, view the runs, etc. Remotely. This allows individuals then to share their data with their collaborators, with other users, and share data more easily. The cartridge itself, I forgot to mention, is 4 capillaries, so we have to say that the instrument itself is not necessarily high throughput, but it is meant for those smaller labs, for those situations where maybe a high throughput instrument is overkill, but you still need to get good high quality data from the gold standard of capillary electrophoresis Thermo Fisher provides. Another question which has come in is what other ways are there to authenticate cell lines besides using STR markers? So there certainly can be done using SNPs, and this is most usually done by NGS solutions. So you would basically generate a SNP profile using next generation sequencing. It certainly is possible to do that, and it provides a lot of data, but in some cases that might just be overkill. Unless you have a very high throughput, sometimes it's more expensive to do this by next-gen sequencing, and you get a lot of data that you might not necessarily need to address the questions you're asking, which is simply that, are these cells what I think they are? So in some cases, next-gen sequencing SNP tag analysis is appropriate. In other cases, the STR analysis is probably a better route to go because it is much quicker, but provides the same level of specificity that a SNP profile might be able to provide. Let's see, in which scenarios do you recommend direct workflow for cell line authentication? So I mentioned in the talk that there are 2 different ways that you can analyze cell lines in human samples. One is by using Identifier Plus, and one is Identifier Direct. Identifier Plus requires genomic DNA for purification for the best, purified genomic DNA for the best results. Identifier Direct can make use of punches on these nucleic cards without having to purify DNA. So my own personal preference is that I like the Identifier Direct workflow because it is so much easier. You don't have to go through a DNA purification step. And also the cards themselves are stable for a very long time, for at least a year. And so you can spot the cards, your cells onto the cards, store them at room temperature for a long time, and then go back to the cards as necessary to repeat or to reanalyze the cell lines from defined periods of time. They also don't take up very much space, and so it's very easy to store the samples on the nucleic cards. Is this workflow applicable for frozen tissue fragments in addition to cell lines? And the answer is yes. Basically, you can do the same workflow to match up any human samples. Again, the kits have been designed to work with forensic samples and to provide a very precise identification of individuals. So if you want to match up human samples, the identifier and global filer kits are ideal ways to go because they provide highly specific fingerprints of the samples and a very, very, very extremely low likelihood that samples from different individuals will have the same STR profile. So it looks like that's all the questions that we have had come in. The webinar itself will be available for download through June 2018, so please revisit the site or direct your friends and colleagues to the site if you need to have more information. And finally, we thank you very much for your time and your interest, and we hope to hear from you soon. Have a good day.